But again, what was really impressive was when they combined ADI with docetaxel, again, you see pretty much the cancer's not growing anymore. So that's pretty much the rationale behind another one of the talks that you heard today, and that was the combination of gemcitabine, which is a nucleoside analog, along with nab-paclitaxel, which is a taxane, and the results are pretty impressive when you take all three of these drugs, ADI plus a nucleoside analog plus a taxane. So we've also looked at a lot of other types of chemotherapies to see whether or not they work well with ADI. And in fact, what we're seeing is lots and lots of drugs work well when combined with ADI, which gives us sort of a problem. And the problem is we're seeing lots of good combinations, but the only way that we're really going to know if these work is to go into clinical studies. And clinical studies cost money and they take time. So we are trying to be very, very selective of which combinations we pursue. All right, so earlier today you heard the talks of some of those ones that we chose to work on, because these are some of the ones that we think are going to give us our best chances. For the rest of the talk, I'm going to be talking about something a little bit different. So yet today, you have not yet heard anything about cancer immunotherapy. And we think that combining ADI with immunotherapy is going to be really good. I'm quite impressed with some of the results that we're getting. So there's been a lot of advances lately in immunotherapy. For instance, it's not too long ago since the drug Yervoy was approved. Yervoy is an antibody that targets a protein called CTLA4, which I'll describe a little bit about in an upcoming slide. Opdivo, which is also known as Nivo, and Pembro, they are also antibodies, and they target this protein on the surface of T cells. And I'll explain a little bit more about that too. But first, a very simplistic overview of the immune therapy or system. So if you start with a tumor, some of those tumor cells will eventually die, and they'll fall into small pieces. And then there is a cell called an antigen-presenting cell. We'll take those small pieces, and it will then present it to a naive T cell. So what a naive T cell is one that has not yet been programmed to attack tumors or anything else. But once the antigen-presenting cell shows it what to attack, it activates it and it becomes an effector T cell. And once you have one effector T cell, it will replicate and produce more effector T cells. And these effector T cells are then going to go off and try and find the tumor and kill it. This is what you want to have, but you also need to have controls in place. And one of the reasons why you need controls in place is that when you have this effector cell and it replicates, you make more of these. And then they can replicate and make more. And they can replicate and make more. And without controls, you could have an awful lot of effector T cells floating around, and that wouldn't be a good thing. So you need to be able to limit the amount of proliferation of these cells. Also something that you need to do is that if this tumor was, for instance, a skin tumor, if it was skin cancer, you want the immune system to attack the cancerous skin cells, but you don't want the immune system to attack healthy skin cells. So there needs to be some way for the immune system to recognize what it is it needs to attack and what it needs to leave alone. So the advances that we're seeing lately have to do with how the immune system is controlled. And it's controlled by checkpoints and T cell exhaustion. So what I want to talk about is this molecule to begin with. So once you have an effector cell and it starts growing, it will also produce on its surface a protein called CTLA4. And so here we have, again, another description of the antigen-presenting cell holding up a piece of the tumor and converting a naive cell into something that's active and can go off and now target tumors. But once it becomes active, it will start producing on its surface a protein called CTLA4. And when it does that, that same antigen-presenting cell that's holding up a piece of that tumor will now bind to it and inactivate it. So it activates when it's a naive cell, it inactivates when it's a T cell because of the CTLA4. The drug Yervo is an antibody that binds here and blocks this interaction. And when you do that, it allows the T cells to stay active for a longer period of time and gives them more opportunity to go find the cancer and attack it. Our drug ends up doing something very similar to that. What our drug does is prevents the CTL4 from coming out to the surface. And if it doesn't come out to the surface, then that interaction can't take place and the T cells will stay active. So here's some data that shows how we came to know that. Here are some measurements of the amounts of CTLA4 that are on the surface of T cells. And over here, what we did was we treated cells with an inactive form of our drug. This drug doesn't work. We intentionally made it so it wouldn't be active. This is a negative control. This bar over here is when we didn't treat the cells with anything at all. And this bar is when we treated with a very, very, very low amount of our drug. Not enough to do anything. And what you see is this is the level of CTLA4 that's being produced. Down here is the level of CTLA4 that's produced when you treat with an appropriate amount of our drug ADI. The less CTLA4 is produced, which means the T cells should now be able to function for a longer period of time and be more active. Now, another checkpoint that you've probably heard an awful lot about these days is PD-1. The way PD-1 works is that it is there to prevent cells, the immune system, from attacking healthy cells. So healthy cells will produce a protein called PD-L1. That PD-L1 reacts with or interacts with PD-1, and when it does, it sends a signal to the T cell saying, don't attack me. Unfortunately, cancer cells have figured this out, or at least some have, and some of the cancer cells produce that same protein, PD-L1, on their surface. And so now when the immune system comes and finds it, it doesn't attack it because the signal is being sent to tell it not to attack. The drugs, Nivo and Pembro, what they do is they're antibodies that they bind to this protein and prevent this interaction from taking place. So when the T cell finds the cancer cell and goes to attack it, nothing sends a signal to it saying don't attack. So it's a great drug. It's being very, very effective in a lot of cancers now. Our drug can do something along that same lines. We measured the amount of PD that was on the surface of T cells when, again, we treated them with the inactive form of our drug, with no drug, with a very, very low amount of drug, and this is the amount of PD that was on the surface of the T cells. But when you treat with our drug, you see less PD. And because you have less PD, there's now less of that signal being sent, and the immune system can do a better job of attacking the cancer cells. There's another regulation that takes place, and it has to do with T regulator cells. What T regulator cells do is they inactivate T effector cells. And how efficient or how strong an immune response is going to depend on what is the ratio between the number of effector cells and the number of regulator cells. When there's a high level of regulator cells, you won't have much of an immune response. When you have high levels of T effector cells, you will. So what we did was measure looked at what our drug does when it comes to regulatory T cells. And what we saw was, okay, here's the inactive form of the drug, here's no drug, here's the very, very, very low level of our drug, and here's the number of T regulator cells that occur when we treat with our drug. Now, so far what I've showed is pretty much ADI makes everything go down. But there's one thing that we don't want to go down, and that's the level of effector cells. So we looked at those. And what we saw was with our drug, here's the inactive form of the drug, here's the no drug, here's the very low level, that's the amount of the T cell proliferation. But then when you add our drug at high enough doses, an increase in the T cells that will attack the cancer. So we are able to shift the T regulator, the T effector, to the point where we have more efficient or a better immune response. Now, we've gone through pretty fast. There's one more thing that I want to talk about, and that is, here is a cartoon depicting a tumor. And tumors, for the most part, are recognized as being something that's foreign, and as a result, the immune system attacks them. And as I've described, there are checkpoints that regulate to what extent the immune system will attack this tumor. And that's what we want, but unfortunately, there are some tumors that aren't immunogenic. And as such, there's not going to be any attacking by the immune system of this cancer. But what we believe is that if you add ADI, it's going to convert the non-immunogenic forms of the cancer into immunogenic ones, allowing the immune system to now attack that tumor. And what makes us think that's going to happen is, what we did is we took a non-immunogenic form, or a weakly immunogenic form of cancer, and we injected it into mice, drew up a tumor, and then extracted the tumor and dyed it with a green dye. And this dye will light up anywhere where there's a T cell. So if the immune system is active, if the immune system is attacking that tumor, what we should see is a whole bunch of green circles. And we don't. But then we did the same exact experiment. Again, injecting this cancer into mice, treating the mice now with ADI, and took them and stained them, and this is what we see. Lots of T cells. This is T cell infiltration. This means that before, with this tumor, drugs like Opdivo and Pembro wouldn't be very effective because there's no T cell infiltration, they're not recognizing it as foreign, so the checkpoint inhibitors won't work. But over here, with the T cell infiltration, you've now created a situation where drugs like Opdivo and Pembro